Okay,
Okay,
so
sir.
let's talk before this session finish. Some trans-acting proteins. The trans-acting proteins, as I explained in the earlier session, that those are the proteins or the elements which came from the other side, which came from the other chromosome, or not from the same stretch of DNA. They are called as the trans-acting proteins or the transcription factors in generally. They are either the transcription factors, number one, or number two, they are certain gene regulatory proteins. So two things, the transcription factors, which directly influence the expression of the gene, and then there are gene regulatory proteins, which can also playing a major role in the expression or in enhancing the expression of a certain gene. So we will going to look into these two things. The transcription factors, generally, you know, the TF2A, TF2B, TF2D, they are like the part of the TI complex, the transcription initiation complex. And that if all these things are there on the promoter region of a gene, the gene will transcribe and it will produce the minimum level of expression, a basal level expression of that gene, a constitutive expression. But if it is supported by the other factors, cis regulatory elements, or with the help of trans regulatory element, the expression level of that gene will be amplified, will be multiplied, will be enhanced a lot. Otherwise, it will going to express at a very minimum or a basal level. The second type of components, the trans regulatory or trans acting components, are gene regulatory proteins. They bind to the specific enhancers, they bind to the specific silencer sequence, and they influence the expression of the genes. I will show you certain examples, and that also include the answer of that original question which I asked in the last class, that why hepatocyte produce factor 8. Okay, so you should dig this thing as well to get the answer of that question. This is just a picture to explain you the difference between the cis and the trans regulation of a gene. For example, there is a gene called gene A. It expresses and produces function, and that gene A is regulated or it is activated by the factor. So this transcription factor is supported with the help of this transcription factor is supported with the help of a long non-coding RNA. This is just an example. What are the long non-coding RNA? We have studied that stuff earlier. So the long non-coding RNA is a supporting factor, and that supporting factor is present on the same stretch of DNA, and that connects and support the factor X to stay on the promoter region of gene A. If this assembly is done, that will indicate that according to our understanding of cis regulatory elements, the long non-coding RNA is acting as a cis regulatory element, putting that factor X in place so that gene A can be expressed comfortably. But everything is present on or coming from the same stretch of DNA. So this is an example of a cis regulatory gene. On the other hand, if a gene is regulated, if the same long non-coding RNA is controlling a gene in a trans manner, the long non-coding RNA will going to express. This is the stretch of a long non-coding RNA, and that long non-coding RNA will bind with another factor, and that factor will eventually proceed to the target gene and then influence the expression of that stuff. So the other gene will be present on another chromosome. The other gene will be present on another chromosome on a far or a different stretch of DNA. So that factor Y can influence the expression of another gene. For example, this is the gene. That factor X can influence the expression of that gene, but that factor Y is activated or supported with the help of long non-coding RNA present on another stretch of DNA. So this is an example of a trans way of controlling the expression of gene. You need to know again, once again, one way or another, you need to know the most of the trans-acting factors, because I don't know which one of them will come in your way. I don't know which one of them you will have to study in detail. So, but I will introduce you about all of them. The first one is very famous, called as CRAB. All these terms initially look very dangerous, very weird, very difficult, but they are like one way or another can be explained in easier ways as well. The CRAB, the C stand for cyclic AMP, R stand for response, E stand for element, and B stand for binding protein. So cyclic AMP, response element, binding protein. So this is a protein which binds to this element. The element is the cyclic AMP. Cyclic AMP response element. I told you in the first class that the DNA element is a stretch of DNA. A certain portion of the DNA can be considered as DNA element if it can bind with a certain thing, with a certain molecule. Like if it can bind with CMP, if it can bind with the CAMP, then this element is called as cyclic AMP response element. And this element can attach, this element can bind with the cyclic AMP, and that is how it can influence the expression of the downstream gene or another gene. So this thing is called as cyclic AMP response element. So whatever the protein which can bind here is called as cyclic AMP response element binding protein. It binds to CRE, and as I showed you earlier, cyclic AMP response element, this one, the CRE, okay? And it can influence the expression of the gene. Usually it is part of the signaling cascade which involves the G protein coupled receptor. And I will show you that what the G protein coupled receptors are. But let me show you if there is any G protein coupled receptor example is here. No, I have shared the document with you, and I will going to open that document so that I will be able to share that how cyclic AMP can influence and can affect the expression of a gene. Okay, please stay with me. I am opening that document for you. It is already placed on your Moodle, so you can download and read. I am assuming that one way or another, whatever the project you will be dealing with, those project needs your understanding that how the proteins and the genes which are assigned to you, which will be assigned to you, are regulated. Okay, so I have 1.5 minutes left. So let me conclude this session. Let me catch you in the session 2, where I will start with the mechanism that how cyclic AMP pathway works and how it is considered as a trans-regulatory element. So this is one of the way that the gene can be influenced. You might be wondering that how many possible ways are out there in order to control the expression of gene. And that gives life the most wonderful form we know. I mean, the complexity behind the life is actually its ability to control in such a tight manner, is such a complex manner. And we are looking at different modes and mechanism. And just imagine if one of them is gone wrong, we can guess the expression of the gene will be affected. And those of you who have studied the PAX gene, they came to know how PAX is involved in the different type of troubles. Okay, so catch me in the second session. Okay, guys, let me proceed without any delay. I'm going to share my screen this time, so I can show you different options over there. Can you see the cyclic AMP pathway document right now?
Yes, sir. Yes, sir.
Okay, cool. Okay, this document is already there, so please, I request you to, I suggest you actually to read this document for your own understanding. So I just show you and share some of the things here. I want to show you how cyclic AMP influence the expression of the gene. So if you can see the things nicely, you can see here. If you can see, here we start. When a cell wants to respond to an external stimuli, most of the time the story starts. अभी people आ रहे हैं, अभी लोग आ रहे हैं हमारे group, अभी और लोग आ रहे हैं. Let them join. Okay. So most of the time.
Excuse me, sir.
जी.
सर थोड़ा सा zoom in कर सकते हैं आप?
Sure. Why not?
Okay, thank you.
Why not? We should. Okay, cool. Thank you. The ठीक है ना? It's better. Okay.
Yes, sir.
So most of the time when we talk about the signaling of the molecule, जब हम बात करते हैं, how a cell responds. Usually cell responds with the help of the receptors present in on their cell membrane. So those receptors have their two types of domains. One domain is present outside the cell, extracellular domain. The other is intracellular domain. Okay, the extracellular domain is the corner of the receptor, which allows the receptor to bind with something. Okay, that something. For example, can you see this triangular thing? This is the something. So that is the stimulus. So if something can bind with the G protein, it's a kind of G protein. It works in couple, so that is why it is called as G protein coupled receptor. So this is the stimulus outside the cell. It binds with the protein receptor, and that changes the conformation in such a way that the intracellular domain is able to do the catalysis. The intracellular domain is able to do a certain type of job. Most of the time it actually phosphorylates something. Most of the time it phosphorylates some molecule. Those molecules sometimes also called as the messenger one. They are messenger, and then those molecule will proceed further and it produce energy, or what I say, it converts ATP into ADP and ATP into cyclic AMP. So the story starts with the entrance of the stimulus. When the stimulus arrived, it activated the G protein. G protein execute its catalysis, and I'm telling you, most of the time it is like a phosphorylation function, and that phosphorylation results in the production of energy or conversion of ATP into cyclic AMP. So that conversion is not possible if this thing is not activated or is not phosphorylated. This thing is phosphorylated once these proteins are activated, and these proteins are activated by G protein, which is then supported or activated by the stimulus. So this is a typical representation of a signaling cascade. But we got the cyclic AMP, and we are studying that how cyclic AMP can influence the expression of the gene. Now this cyclic AMP can contribute, can change, can convert into different forms and convert into different things depending on the nature of phosphorylation. And in the meanwhile, it converts the pKa. It's a kinase. It converts a protein called pKa. It made it activated, and most of the time in cell signaling, activated means phosphorylated. It phosphorylates the protein, convert the inactivated version of pKa into an active version of pKa. So everything depends on the presence of CAMP here. So the CAMP indirectly pushes a signaling cascade which leads to the activation of pKa. Now that pKa can enter into the nucleus, can pass through the nuclear membrane, and then it can bind to the target genes. It can bind to the specific locations, and it can bind with the specific proteins, take those specific proteins to the target sequence of certain genes, and those genes will be expressed. So one of the way that cyclic AMP can influence the expression of the gene is that cyclic AMP can activate pKa protein kinase, and those protein kinases, they are able to cross the nuclear membrane, and they can activate the downstream proteins which can bind to the specific target sequences and trigger the expression of certain genes. So that's the one of the way cyclic AMP can send activated molecules within the nucleus so that they can influence, they can affect the expression of genes. What we are studying here is that there exists a cyclic AMP response element. I wish I able to make sense. There are cyclic AMP response elements present on the DNA. Remember, this is the DNA stretch. This is the DNA. And that DNA contains a cyclic AMP response element. A certain stretch of DNA can sense the cyclic AMP, can bind with the cyclic AMP. So all those proteins which can bind here, they can influence the expression of those genes. From where those proteins are coming? Those proteins are coming from different locations, and all of those locations are involved in the regulation of that gene. So for example, this is the location 1, this is the location number 2, this is the location number 3, but all of these 3 proteins can recognize, can bind with the cyclic AMP, or more easily, that part is called as the CRE, cyclic AMP response element. So all of these 3 proteins can influence that DNA element. That means all of these 3 proteins can influence the expression of the gene as well. So one gene at the same time can be influenced by multiple partners coming from the different locations. And all these 3 different components, or 4 components, or 5 components, or 1 components can influence the expression of gene. So this is one of the way. The other type of components are serum response factor. Serum response factor can bind to a DNA element called SRE. Similarly to this one. This is the cyclic AMP response element. Similar type of element exists on our DNA, which is called as SRE. The SRE elements are the ones which can bind serum response factors. And they're usually present in the serum. They are mostly growth factors, and they can induce the expression of certain genes as well. And then there are STAT1, then there are MAP1, and there are AP1 binding sites. These are different response elements, and they work in the same way. But you can see that they are influenced by different factors. For example, MAP1, it binds to HMRE. Remember, the RE is the response element. So HMRE is the heavy metal response element. It can respond to the heavy metals in the cells. If the heavy metals are there, for example, let me share. So this is a gene which is not supposed to be expressed in a normal conditions. It is controlled or regulated by a DNA element, and that DNA element is called as HMRE. HMRE means this is a heavy metal response element. This gene will not be expressed in normal condition until unless there is a heavy metal out there. Until unless there is a heavy metal over there. This heavy metal can bind with the heavy metal response element and then activate the expression of that gene. And of course, the cell will respond to the new situation after that. Now, the heavy metal is coming from outside. It is coming from away. It is coming from, not coming from the same DNA stretch. It is coming from somewhere else. So it's a trans-regulatory factor. It's a trans-regulatory element. So that is how this thing can be considered as the trans factor. But in the normal conditions, this gene is not required to be expressed, because this gene only work or only required to be expressed in a terrible conditions when you have heavy metals in your genes. For your brainstorming, okay, there are other more type of similar response elements exist out there. For your brainstorming, I am just telling you, and you can go back and study this type of things just for your brainstorming. Let's suppose that there is a gene and we have discovered a response element. We have discovered a response element which can get activated, which can bind or get activated in the presence of carbon dioxide or in the presence of carbon monoxide. Let's suppose in the presence of carbon monoxide. So this is called carbon monoxide response element. Okay, it makes sense. hypothetically, if I'm telling you that there is a DNA element which can respond to the carbon monoxide. So this DNA element will be considered as carbon monoxide response element. So if the carbon monoxide is not there, that gene is not required. The gene will not express. But what if a carbon monoxide gas is there? A carbon monoxide gas is there that will going to activate this DNA element. The DNA element is called as carbon monoxide response element. And what will happen? That gene will be expressed. If you install that gene into a bacteria and put green fluorescent protein or GFP here, put a green fluorescent protein or GFP here, what will happen? In the absence of carbon monoxide gas, the GFP will not express, the bacteria will not express any GFP, but in the presence of carbon monoxide gas, that DNA element will be activated and that will trigger the expression of GFP and the bacteria will grow in green color. So that glow indicates the presence of carbon monoxide gas in the system. Hope you are getting my point. So that is how many people in the synthetic biology, they are looking for the DNA elements which can respond to the elements of their own choice. For example, methane response element should be there, which can activate in the presence of methane. Heat response element should be there, which can activate in the presence of heat. Cold shock response element should be there. So you will see certain types of elements are there, which helps our cells to respond to certain weird type of situations. So somehow, whether we are literate or whether we are illiterate person, whether we study anything or we study at all, our cells understand that in the presence of different type of factors, different types of genes are required to be expressed. So people are looking for these response elements to create things which can respond in different conditions. Like they want to create bacteria which can respond when the carbon dioxide level gas increases. So they can monitor carbon dioxide gas with the help of those bacteria, or carbon monoxide gas or any other gas if they found a response element. Okay. There are other proteins, they are regulatory proteins, okay, trans-regulatory proteins coming from somewhere else and controlling the expression of genes. A little boring, but they are very important, okay? One way or another, you need to come back and you need to see whatever is your assigned project, that protein or gene is belong to which category. So you came to know how it is regulated. That is a very important information you should have. Because if you understand how it is regulated, you can work that how it get dysregulated in a certain disease. So let's start with the first type of DNA binding proteins called homodomain proteins. They are very famous one. Homodomain proteins, they are small, but they are doing a wonderful job in our systems. Do I have a question in the chat? Not yes, yes. Okay. The homodomain proteins, they are consist of 3 alpha helices. Okay, there are 3 alpha helices are out there. Alpha helices, 3 alpha helices. You can figure it out. The one is very clear. You can see the white one. If you can see my mouse, you can see the white one here. Let me highlight the. So the one of the alpha helix you can see in the white. The other one is here, and the third one is you can see here. So there are 3 alpha helices here, and then you can see there's 60 amino acid homodomain present here, and that's why it is called as a homodomain protein. It consists of a region, and this region is responsible for its binding with the DNA. Remember, every DNA binding protein, and this is another discussion, I have already gone through that thing, that presence of DNA itself is nothing. Every stretch of DNA is loaded with different types of proteins, and those proteins control the expression of the DNA, and those are called as DNA binding proteins. And one of the DNA binding protein is called as Homodomain proteins. And there are 60 base pair, this region, there are 60 amino acid region is involved in the binding of the DNA. For example, there is a gene called GH. It's a growth hormone. It's a very important gene, growth hormone gene. Can you see this thing? Like a tail stuff, something here. This is a homodomain protein called PIT1. It's a PIT1, very famous homodomain protein. It can bind. So this is the trans TI complex. Am I right? The TI complex, where at the core promoter region. So everything is set up. The core promoter, the TI complex is there. It will give you a basal level of expression of growth hormone. That will give you a basal level of growth as well. But the PIT1 homodomain protein, it can bind here with that DNA, and it's binding with the help of the 60 amino acid homodomain here with the DNA, and that will activate the T1 complex a lot. And that is why it can run faster. It can trigger the expression, many folds more than the basal level. The presence of this PIT1 protein will trigger the expression of growth hormone, TSH gene, and it affects the growth a lot. So that is how it influences the expression of the thing. Without the PIT1, the growth hormone or TSH will going to express in a constitutive level. Any bad or any mutation in the PIT1 gene, what will happen? So this is the PIT1 gene. Let's suppose something bad happen here. If you are working on, that's for example, your research project is on PIT1. So if a mutation here on the PIT1 home box or homodomain region, home box region produce 60 amino acid homodomain, and that is the portion with the help of which that protein is able to bind with the DNA. So if something bad here, that will going to influence the structure of that protein, and that protein will not be able to bind with the DNA. If that will not able to bind with the DNA, although the growth hormone is perfect, although the TSH is perfect, although the RPL gene is perfect, but because of the absence of this binding, due to the mutation in that gene, the growth hormone will not going to be expressed in an optimal level. It will always stay at a basal level, and that trigger and that affects and that cause the disease, and that results in the deficiency of growth hormone and the deficiency of thyroid stimulating hormone and the deficiency of PRL, which is prolactin, and that called less growth or dwarfism. जो dwarfism होता है लोगों में, that is because of this thing. So this thing is perfect, but because of the error here, it will lead to the overall decrease in the growth factor and cause such a big disease. Okay, so you can think about a project using from here, but you need dwarf people. You need dwarf people to get the samples to find out this. Okay. Oh, मैंने everything desire. So this is a homodomain protein, and this is the that how PIT1 regulate the expression of the growth hormone. So you know that here is the at the promoter region, at the core promoter region, the TI complex is here. That complex is not good enough until unless it is supported by this PIT1 protein. The PIT1 binds here and it support, and that results in the production of growth hormone. And then that growth hormone produce, go out and execute the function. Okay, so that's it. Okay, from this, let's move to the second part. साथ में रहना, okay? थोड़ा boring है लेकिन साथ में रहना. The second type of DNA binding proteins, we are talking about the DNA binding proteins called Leucine Zipper proteins. The Leucine Zipper proteins, they consist of an alpha helix in such a way that every seventh amino acid is leucine. Can you see? Every seventh amino acid on this alpha helix is a leucine. Leucine. And Leucine zipper proteins occur in dimers. So these two leucines allow these two proteins to dimerize with each other in this way. They allow them to dimerize. So this is the leucine zipper protein called June. This is another leucine zipper protein called June. With the help of their properly aligned leucine, they can dimerize like a zipper. They can dimerize. And this dimerization leads to a Y-shaped protein. And you can see this portion, the lower part of that leucine zipper, it gives that protein an ability to bind with the specific part of the DNA. So this 20 amino acid portion here, somewhere here, 20 amino acids here, 20 amino acids here, will give DNA binding domain. So that is how it can bind, it can connect with any DNA. The special sequence of DNA it can bind. Now, the thing you need to know, the dimerization can occur between two similar zipper proteins like June and June. Homodomer, it is called homodimer, and the dimerization can be done between dissimilar leucine zipper proteins like phos and June. It is between phos and June. So there are different types of leucine zipper DNA binding proteins which has their defined targets, and it can influence and brings the change in the expression of target genes. These are the examples of the leucine zipper proteins and their targets. Some of them are very important, and I wish that you people should proceed and go and see that how they control the expression of these different type of genes. Or maybe when you will be given with a project, the protein which you will be working on will belong one of these categories, and you can guess any dysfunction in those proteins will affect the expression of the target genes. So let's suppose this is the target gene, and this target gene is influenced or regulated by a leucine zipper. Any bad, for example, phos protein, if the phos protein is not there, if the gene which is producing the phos protein is not working, so this is the gene which produces the phos protein. If anything bad happens to the gene, the phos protein is not there, and ultimately it will not going to influence. It will not able to influence the target gene. So the expression of the target gene will not be there as well. It's a proto-oncogene. The phos gene is a proto-oncogene. I will explain you the oncogenes and tumor expression in the next lecture. Okay, so that is how June is a very famous protein as well, controlling a lot of wonderful things. It's a proto-oncogene as well. Remember, the proto-oncogene is not the bad thing. It's not a bad thing. It seems weird. It spells weird, but proto-oncogene is a gene which is required by the cells to proceed with the normal functions, to proceed with the normal cell divisions. But if something bad happens, soon आप microphone off कर दें. If something bad happens with the proto-oncogene, if proto-oncogene is responsible for the cell division in a regulated way, if the proto-oncogene is not working, the function is lost. So the loss of the function in the proto-oncogene will convert that proto-oncogene into an oncogene. Convert them into an oncogene, and that's why it can cause the cancer. तो इसके नाम के अंदर भी proto-oncogene आ गया जिससे बुरा लगता है, लेकिन it's an important thing required for the normal function. You can consider that thing in an analogy like this. That for example, the accelerator in your car is a proto-oncogene. If you ask me, the accelerator is required for the normal function of the car, the answer is yes. We speeds up with the help of accelerator when it is required. But if something bad happens to the accelerator, the same thing which gives the normal function, the same thing might be a reason of trouble. If we able to push the accelerator and we are not able to relax it back, it kept on accelerating the car, that will lead to an accident. So that is how something which can push the cells for the cell division, can be a reason of trouble for the cell as well. So those things are called as proto-oncogene. Do not consider them bad. They are required, but if they get bad, they will convert it into oncogenes. Okay, so here we go. The next one is the helix loop helix proteins. नाम से जाहिर है, they have helix, loop, and then helix proteins, and they are also present in dimer. They have a short alpha helix. They have a very short alpha helix here, okay, connected by a loop. So this is the alpha helix, and then this is the loop, and then there is a long alpha helix here. So helix, loop, and helix. So that is why it is called as helix loop helix. And they also exist in dimerized form. The two proteins join with each other. Once they join in this fashion, in this way. It's not like Leucine Zipper. They dimerize to create like a scissor, something like that. The dimerization can be done between the same proteins, homodimers, and the dimerization can also be done between different proteins called heterodimers. Okay, the famous, very famous, MyoD and MYC. Both of them are very important and very famous proteins. Both of them belongs to the helix loop helix protein category. They regulate many important genes. The MyoD, they regulate the genes which involve in muscle development. So guess what will happen? You need to give me the answer. If MyoD is not working, what will happen? क्या होगा उसके नतीजे में? Guess करें. Tell me क्या होगा?
Sir, skinny person होगा like me.
Okay. Anybody else?
मसल एट्रोफी हो जाएगी सर.
क्या होगा?
सर डवार्फ तो नहीं हो जाएगी?
Okay. और
सर मसल एट्रोफी.
If a MyoD, which is supposed to regulate muscle development, the MyoD is not working, क्या होगा? शाबाश. आप लोगों का कंसेंसस हो गया इसके ऊपर जो आपने मुझे बताया है? क्या हो गया? ठीक है. ओ रिजा ने टाइप किया मसल डिसइंटीग्रेशन. ओके, शाबाश. मैं आपके लिए ना गूगल कर रहा हूँ इन द मीनवाइल, आपको दिखाने के लिए. ओके, ये क्या होगा? दैट माइट बी मोर. किसी ने ये जीन जानबूझ के निकाला, ठीक है, जानबूझ के निकाला एक एनिमल में से. किसी भाई ने जानबूझ के ये जीन निकाला और मैं आपको वो साहब दिखाता हूँ और साथ में वो एनिमल भी दिखाता हूँ. ओके, सो यू कैन गैस कि किसका आंसर ठीक होता है. Here we go. किसका आंसर ठीक हुआ? किसी ने MyoD को डिलीट कर दिया. कैन यू सी द बुल?
यस सर.
Oh my God. तो फिर ये स्किनी ना हुआ ना. तो if nobody is there to control the function of muscle development, you can guess that the muscle will grow out of control. That is not nice, by the way, but people want to have it. Okay, the next is G. The next is that the MYC protein is also very important. It regulates brilliant and important functions. It's encoded by the MYC gene. It's a proto-oncogene. So now you should know what is proto-oncogene. VMYC, it present on this chromosome, and you should know in the same way about the MYC as well. Okay, बहुत आपके सामने आएगा बार बार. The next category, okay, बस हिम्मत और बहादुरी से रहना, ठीक है, is called the zinc finger proteins. They are another DNA binding proteins. The special thing about the zinc finger protein is that they contains it is not possible for me to show you here, but you can zoom in later on, download the slide and see. जहां मैंने circle किया है, You can see a zinc in the center. The zinc finger protein contains a zinc, okay, and that zinc is connecting different domains of that proteins, like here. Zinc can connect the different domains of that protein. That is why it is called as the zinc finger protein. It is involved in the basic shape of that protein. It's a DNA binding protein. It binds with the DNA with the help of a certain DNA binding domain. This is the DNA binding domain I am coloring right now. So the DNA binding domain allows the zinc finger proteins to bind with the target DNA. The important thing about the zinc finger DNA binding protein is that people have engineered the zinc finger domains or zinc finger proteins to send the nucleases or DNA cutting tools at the place of their own choice. People used zinc finger domains, zinc finger proteins, to send the cutters at the place of their own choice within human genome. It was an obsolete technique, but it was the best technique of that time in 2010, 8, and 9. After that, we discovered Talons, another level of targeted genome editing, and nowadays we are using CRISPR-Cas9 system to target a place of our own choice on our genome. So remember, the zinc finger proteins are forefathers of the targeted genome editing. But this is not the topic here, so I'm not pushing it further. I'm just explaining you that the zinc finger zinc containing protein, it can bind with different molecules, and in this example you can see it can bind with a steroid, and this is an example of glucocorticoid receptor. So this is the glucocorticoid. So a zinc finger can brings in the presence of this steroid. If the steroid is there, it can bind with the zinc finger protein. Zinc finger protein on the other side have a DNA binding protein, and it can bind with the target DNA. It brings that steroid to that place. So that is why it can act and support the expression of that gene. So this is one of the way, as I showed you one example only, the glucocorticoid receptor, that one zinc finger, how it can influence the expression of the certain genes by responding to steroids or any other chemical. So the TI complex is there, it will express at a basal level, but because of this zinc finger protein here, it can increase its expression to many folds. And that zinc finger is only able to bind here once it receives that glucocorticoid, but it receives that steroid. It activates or it binds with the zinc finger protein, allows it to bind it with the target site and increase the expression of that gene. So that is how it works. It has more things to explain, but that's enough for today. And then we know about the microRNA. I have explained that how microRNA regulate the expression of gene already in detail. And then the antisense, how antisense genes can express and control the expression of genes. We have studied it in detail, no problem. And then the riboswitch. The riboswitch genes are the genes which can be controlled because of its structure. Let's see how. So this is the DNA here. This is the DNA, and this is the riboswitch gene. So the riboswitch gene can control its expression because of its own structure. See how. So once a riboswitch gene is transcribed, it will produce a messenger RNA, riboswitch RNA, that will go into the cytoplasm. And because of its sequence, it folds into a definite three-dimensional structure. This is an inactivated version of a riboswitch RNA. That RNA cannot be used by the ribosomes to produce protein. So it will not be expressed. It will wait for an external support of a target molecule which can fit into one of its domain. So based on the shape of the riboswitch RNA, the external molecule will fit itself into the riboswitch, and that allows that riboswitch to RNA to convert from inactivated version to an activated form. So with the help of this target molecule, the riboswitch RNA can convert itself from inactivated version to an activated version, and now ribosome can detect it and can produce the target protein. So you can guess the target protein can be regulated just because of the structure of RNA. So RNA is there. If a molecule is not there, it will not be converted into activated form. It will not going to produce any protein product.